Welcome everybody. So this is our final meeting on the application of bacteriophages in biotechnology, and today we're going to talk about how to use phages for food preservation, fight agriculture diseases, water decontamination, and corrosion control. So let's begin. If we talk about biocontrol, I want to remind you what we were talking about on our previous meetings. So we were talking about using phages, for example, like to treat animals that are used in farms, for industrial sanitation of different production areas. This also influences the products. To use phages to treat raw products, like to prolong their freshness, to get rid of pathogenic bacteria that can be on these raw products. And of course, we are talking about phages to use them for preservation of ready-to-use products in markets and those products that come right to consumer. And also, just a little remark, not only phages can be used, but also some parts of the phages. They are called bacteriocins. Bacteriocins, it's a group of substances. Usually, these substances that are called bacteriocins are produced by bacteria to kill other bacteria. Antibiotics are produced by fungi, and they kill bacteria. Bacteriocins are produced by bacteria, and they kill bacteria. And sometimes these bacteriocins, these substances, are parts of viruses, like for example phage tails. And these phage tails also can be used in food and health in different areas, like phages to fight pathogenic bacteria. So these bacteriocins and phages can be used, for example, for pathogen reduction and to fight plant diseases. Because we must remember that animals, they also have to eat some food, and if we talk about plant food, this plant food also must be free of different pathogenic bacteria, and phages can help us to do that. If we talk about already products, so we can also use phages for preservation of these products, to keep them fresh, to prolong their shelf life, to keep them fresh for a longer time. Also, we talk about phages for biosanitation. It means that we can treat different production areas and surfaces with phages instead of chemical compounds, so that also to get rid of pathogenic bacteria. If bacteria will be present on these areas, they can infect products. If we will get rid of these bacteria, our products also will be safe. And we talk about food safety and preservation using phages instead of different chemical conservations and different antibiotics that can be used. So we can simply use phages to eliminate pathogenic bacteria from products. Phages can simply kill bacterial cells, or we can use only phage enzyme endolysin. This enzyme is used by phages to break through the cell wall of bacteria, and so we can simply use this enzyme also to kill bacterial cells. And we can all use everything to preserve food. But we must remember that phages can be not only the answer to the problem, but the source of the problem. If we talk about phages that kill pathogenic bacteria, we talk about the salvation of the problem, we talk about preservation of food. But there are some types of food that are called fermented food. Food fermentation is a process where special bacteria are used. For example, to create cheese, we need some special bacteria, and if at the moment of this fermentation process, a phage specific to this bacteria will get into this fermenter, so the fermentation process will fail, and it will be stopped, it will be ruined, so the phage becomes a problem. But anyway, people try to take everything they can from any process, and now these phages, for example, can be used in wine making. If we talk about fermentation, we use special types of bacteria to get some special properties and qualities of the product, fermented product, and for example, for the special taste. And sometimes we have to get rid of some bacteria and keep other strains of bacteria. So phages can be used because of their specificity to certain bacteria to get rid of unwanted bacteria and to leave the bacteria of interest in the fermentation process. When we apply phages on food, we must remember that there are some also problems to be solved or questions to be raised, because we need to know when to apply the phage, how much phage must be applied, so the amount of the phage. We must know what phage to use. Maybe we have to make some changes in the phage structure. Maybe it has to be edited, like biotechnologically changed. We also have to know how to apply the phage. Maybe it can be simply applied to food, or maybe it's need carrier, and whether at all can we use the phage to this particular product, or we just can't. And also the temperature question, because sometimes the temperature for the product must be too high or too cold, but it is a problem for a phage because it won't work when the temperature is too high or too cold. It will just stop, and we won't get any effect of using phage if the temperature isn't optimal for the phage himself. So what I'm trying to say is a very difficult process to find the right phage to use. It's not just simply we take the phage from the environment and we use it for food preservation or to, to resolve some problems. We have to find the right phage to use. Then sometimes it's needed some genetical design, something has to be changed to make this phage better for the process. Maybe we can even rebuild it a little bit, the proteins of the phage. Then the phage must be tested, whether it's safe, whether on its efficacy, whether it's do we really need to use this phage in the process? And then we can only use it for preservation of food and for treatment of domestic animals. And I'm not even talking about medicine. Of course, medicine also needs all these stages. So, agricultural diseases, plants like animals and human also can get ill, can get sick, and the agents that cause these illnesses are viruses, plant viruses, phytopathogenic bacteria, and fungi. So the problem of bacteria can be solved in the ecological way using phages, because phages are just part of the environment, and we can simply put a larger amount of the phages where we have our plants. And so the phages will fight phytopathogenic bacteria like Erwinia, Ralstonia, Xanthomonas, Pseudomonas that cause the diseases of plants. And we must remember that every time the phage infects the cell, the new generation of phages comes out and also can take control of these phytopathogenic bacteria. When we talk about biocontrol of bacteria using phages, we must remember that this is a natural system where every time the bacteria tries to escape the infection, the phage finds its way to infect the bacteria. And every time the bacteria finds a new way to infect the plant, the phage anyway will find its way to fight these bacteria in nature, because this is absolutely natural process, because biocontrol is the idea that we use something biological to fight some biological problem. And the system bacteria and phage is a really good system to be used in biocontrol. When we use phages to treat plants, we must also remember and take into the account where the infection takes place. If we talk about the root system, if bacteria infects root system, for example, we have to put these roots to the suspension containing phages, or we can use other ways to put the phage to the soil, whether we can pour it, or if we talk about the infection of leaves, so maybe we sometimes even have to spray the phage simply on the plants, and we will have the effect of the healthy plant. And we mustn't forget about using phage cocktails. So using a lot of phages at the same time, like phage cocktails, also will help us to prevent the disease, because here can be phages that are a lot of different phages, one and the same bacteria. If it becomes, for example, insensitive to one of them, the others will work. Or here can be different phages to different types of bacteria, because a lot of bacteria can simultaneously attack a plant, and we can help to solve the problem using simultaneously a lot of phages in the phage cocktail. If we talk about water decontamination, usually all the different filters we use, they help us to eliminate physical and chemical pollution, like different dust parts, dirt, like some chemical compounds in this water. But how to get rid of the pathogenic bacteria present in this water? Because sometimes bacterial cells can be eliminated like physical pollution, but anyway we need some chemical compounds to kill these bacteria pathogens. It means that we clean the water from one group of chemicals and we use other group of chemicals like antibiotics and different disinfectives to get rid of the bacteria. But there is a more natural way, like to use phages, and phages can be used, for example, for wastewater decontamination to control the amount of pathogenic bacteria in the wastewater that comes to the environment. And also we can use phages when we take the water from the environment, for example, also to monitor the harmful bacteria or to just track the pollution, because different types of phages can be used in this water decontamination system. For example, like to find the source of infection. They help us to detect if we don't know where the pollution comes from. So phages can be used, and also phage-based biosensors. We were talking about biosensors. They also can be used. And when we talk about phages and water decontamination, there is also one interesting thing about what bacteria do to escape the infection and escape treatment with antibiotics. So we talk about water collection systems, and we talk about water distribution systems where bacteria can form biofilms. Biofilm, it's not just a group of bacteria in the sort of slime. Biofilm is an organized structure where bacterial cells have their own functions, and it's not that easy to break through the biofilm. So the biofilm becomes a problem, because if we have the biofilm in the collection systems or distribution systems, this is the source of continuous contamination of water with bacterial cells. And it's easy to kill one bacterial cell if we know how to kill it, but it's really difficult to get through the biofilm, because bacterial cells are unreachable for antibiotics, for chemical compounds, and these biofilms are a problem also in hospitals where they've super resistant bacteria, they form biofilms and they can infect patients in the hospitals, and biofilms become a really serious problem. And talking about biofilms, we also have to talk about corrosion. So corrosion of metal can be caused not only by physical or chemical factors, but also by bacteria that produce some substances, and they cause the spoiling of metals. And this is a really serious problem for different constructions. And bacterial biofilms are also a problem with metal corrosion, because these biofilms are completely unreachable for different compounds, different antibiotics, as I already said. But phages can help to solve the problem, because phages have some sort of special enzymes that help them break through the biofilm and to infect at least one bacterial cell. And then the chances to get to other bacterial cells in the biofilm are much higher. So the composition of chemical compounds, maybe antibiotics and phages all together help to break biofilms and to solve the problem with the contamination of different water systems, with biofilms in hospitals, and of course with biofilms that cause the spoiling, the corrosion of metals. So we were talking about using of phages in different areas in medicine, like to an alternative to antibiotics, for drug and vaccine delivery, for sanitation of different surfaces and decontamination of different surfaces, to using phages to treat animals and to treat plants, and also to break biofilms. But we must remember that every time we use something biological, especially, we must remember about safety. So biosafety is a key question, because phages can be our friends or our enemies. They help us in phage therapy, like alternative to antibiotics. Phages help us because we can change their properties genetically and also use them in different areas. Phages help us to solve the problem of biofilms. Phages help us simply detect or change bacteria also genetically. But phages can also become a problem, because if the bacteria is resistant to a phage, well, this problem can be easily solved, but anyway, this is a problem, the resistance of bacterial cells to infection with phages. Phages can increase biofilm production. It's like a fight back from the bacteria, like an answer to the tries to break the biofilm, so the bacteria answer with making even overproduction of these biofilms. Also, phages, you remember that phages can cause not only lytic reproduction cycle within bacteria, but also they can cause lysogenization of bacteria. And phages, when they build their genome inside the genome of the cell, they can also perform their horizontal gene transfer, and they can also bring to the cell genes of resistance to antibiotics. So it's like on one side we use phages to fight antibiotic-resistant bacteria. On the other side, phages can bring the genes of resistance to bacterial cells, and these bacteria also can become resistant to antibiotics. So it's like a life cycle. But anyway, this problem has to be maybe not resolved, but we must remember that phages are biological systems that have different sides of their life, let's call it this way. So, in the conclusion, what you already know and what I want you to remember from all the topics we're talking about. Phages, bacteriophages, are viruses that infect bacteria. They are safe to animals, plants, and humans. They can be used in biotechnology. Phages have their protein coat and genetic material. Usually this genetic material is double-stranded DNA. Phages have head, tail, and tail fibers. Positive sides of using phages. They are efficient as antimicrobial agents, alternative to antibiotics. High selectivity of phages. They are very specific in their interactions with bacteria. Phages can help us in the natural way to escape bacterial resistance. Even if bacteria becomes resistant to this phage, it's absolutely natural process for the phage to find a new way to infect this bacteria. And phages are very cheap in their production. So these drugs based on phages are really very cheap. But there are also negative sides, because even if bacterial resistance can be solved, there are problems with preparation and administration of bacteriophages, because it's really important to deliver them right to the point where they are needed. But we remember that phages can activate the host immune system. The immune system of macroorganism can answer to the proteins of the phage and eliminate the phage before the phage reaches bacteria. And phages, bacteriophages, drugs based on bacteriophages are not patentable. It's really difficult to patent something that is absolutely normal biological cycle. The lifestyle of the phage cannot be patented. So it's absolutely natural for a phage to infect bacteria and kill it, and you cannot make a patent on it. And you remember that we talked about phage cocktails. This is a combination of different phages that can infect either one bacteria or different bacteria. So it's like a combination of drugs, phage drugs, to enhance the effect of treatment. Also, phages can be, we can use not only phages, but their parts, like their enzymes, can be also effectively used to destroy bacterial cells. Phages and antibiotics altogether can enhance the effect of treatment if we talk about some serious infectious diseases. We can also use engineered phages. We can change the protein structure of phages, or we can change the genome of phage, the genetic system of the phage. Also, we can make phage a carrier of new proteins, like a vector. And we remember about CRISPR-Cas systems, also connected with phages, because these systems, CRISPR-Cas systems, gave us the ability to make changes in the genomes of different organisms and to resolve the problem of diseases. So these CRISPR-Cas systems are also very effective. And so I think that this will be enough for today and at all for this topic. It's not everything you can find about the application of bacteriophages in biotechnology. So I hope that this information was useful and interesting for you, and that you will look further for all the other materials and maybe some new materials coming out right now in the scientific world. So I thank you for your attention, and so this is it. Thank you and goodbye.